stimulus unmasks positive feedback in an auto-regulated bacterial signaling circuit. It was published in the Proceedings of the National Academy of Sciences in 2008. So their work examined the FOQ, FOP two-component signaling system, in which FOQ is the sensor which senses extracellular conditions and activates the FOP response regulator. The FOQ sensor is a bifunctional histidine kinase, which means that it both phosphorylates and dephosphorylates the response regulator FOP. And really the only other significant thing they knew about the system was that at some point autoregulation affects response regulator activation. They didn't know the exact details of how this feedback is propagated. They knew that it occurs, and so this led to their formation of two central questions of when does feedback amplify the signal. Feedback is usually not something that's constant across ranges of input, but rather it's stimulus dependent. And then they sought to develop a model to explain this and ask how does the system control stimulus dependent feedback. And hopefully with the development of the model, they can make predictions about stimulus dependent feedback. So they also knew the kind of genetic background for how autoregulation occurs in this system. Besides the usual two-component signaling cascade of FOQ activation from some environmental signal being propagated by autophosphorylation to phosphorylate FOP response regulator, FOP activates response targets, but additionally activates the FOP-FOQ operon. And this operon codes for FOP, FOQ. It is normally driven by constitutive promoter P2, but it's also driven by this phosphorylated FOP dependent promoter, which they term P1. So it's this P1 induction which closes this feedback loop and causes autoregulation. So in order to examine how autoregulation affects system output, they engineered a strain of E. coli with autoregulation deficiency. They did this by knocking out this promoter P1 in the P1 minus strain, and they use that and compare it to the P1 plus strain, which acts as a wild type of normal autoregulation. The first thing they examined, levels of FOP, the response regulator, and they determined that in the wild type, autoregulation does occur with high stimulus. Magnesium response to FOQ, FOP is inverted, so low magnesium is a high stimulus, and you see there's an accumulation of FOP response regulator. In the P1 minus, there's no such accumulation, so it suggests that lack of autoregulation inhibits this FOP production, and the P1 promoter is not induced. But FOP is the response regulator, remember, not the system output. So then they sought to determine what the system output response was. And they used this by fusing YFP to one of the system output targets as MGRP gene. They compared that to a constitutively expressed CFP as an internal fluorescence standard. When they did this, they were surprised to see that there's absolutely no effect from autoregulation across this wide range of stimulus. Across this range of magnesium concentrations, the P1 plus and P1 minus strains have almost identical system response. In order to determine why this is happening and hopefully predict where the feedback actually comes into play, they developed a systems level model. And this model started off with breaking the system into two modules. The first module is the standard two-component signaling system. Their inputs of stimulus and total FOP and FOQ with an output of phosphorylated FOP. And this sort of signaling system diagram can be represented also by an input-output curve, where the input is on the x-axis and the corresponding output on the y. And implicit in this, these curves is the assumption that the output is saturated with increasing input. And analysis of other systems suggests that this be the case. The second module their model was the auto-regulation module. So this would be the input being phosphorylated or activated FOP, and the output being more FOP, FOQ, driven by the expression of the FOP, FOQ operon. For this input-output curve, there are two scenarios depicted. For basal expression, in which there's no autoregulation, the concentration of FOP is constant. There's nothing changing the operation or the activation of the FOP, FOQ operon. But under autoregulation, there is an increase in concentration of FOP corresponding to increases in phosphorylated FOP. So what do you do when you have two modules? You stick them together and link the inputs and outputs of the two different modules. And you can use a trick from systems engineering, in which you actually overlay the input-output curves in the two different modules, and you can look at intersections of these curves to determine what's called steady-state behavior of the system. We can see how the system will function under various stimuli and presence or absence of autoregulation. So the first module, input-output curve, and the second, you can see the axes are a bit swapped, so with a little bit of trickery and reflection, rotation, and translation, you can see the overlay. And the important takeaway here is that under conditions of low stimulus, there's very little difference in phosphorylated FOP system output based on presence or absence of autoregulation. This intersection represents the system with autoregulation, blue without. However, at high stimulus, there's a significant difference in phosphorylated FOP levels between the autoregulated and the non-autoregulated system. So from this model, their authors were able to predict that the way to expose this system's feedback response was to analyze it under very high stimulus. And very high stimulus in the FOQ-FOP system corresponds to very low magnesium levels. So what they did was take those P1 plus and P1 minus strains and grow them under growth-limiting concentrations of magnesium, so basically the lowest concentration of magnesium possible. And voila, they saw an incredible difference between feedback in the autoregulated strain and the non-autoregulated strain. So finally, they had exposed where feedback was propagated in the system. And you can kind of follow the systems level analysis of the difference between phosphorylated FOP in the non-autoregulated system and the autoregulated system, leading to increase in protein target regulation and the increase in system output. Their analysis went back to their central questions. When does feedback occur? The model predicted, and the following experiments confirmed that high input is one way to expose feedback in this system. And their question of how is feedback regulated is answered by this unsatisfyingly complex but very useful model of how phosphorylated FOP levels are adjusted based on the presence or absence of autoregulation. Understanding the system is rather important. It's found in the FOP-FOQ system is one member of a family of two-component signaling systems in bacteria. So understanding this provides some questions to a bunch of interesting biological questions about how bacteria respond to changes in their environment. If we understand all the family of different responses, we can understand why the bacteria might want to respond to a certain environmental cue. But also from the perspective of a systems engineer trying to build biological systems, this sort of autoregulation provides a tool for the biological engineer to modulate dynamic range in these two-component systems. So it provides a way to have a steady, robust, consistent system output for majority of inputs, and then really trigger at a high stimulus. So thanks to my instructors, Natalie Coldell, Tyler DeWitt, and happy to take questions.